Hi everyone. Today I'm going to be talking about transduction of mechanical waves and electrical impulses, which is basically how we can hear and how we can turn sound waves into action potentials. So hearing depends on a series of transductions, which is it means to convert one form of energy into another form of energy. So sound energy gets turned into fluid waves and then into movement of hair cells and then finally into action potentials that the brain can read. And there are properties of sound that are important to understand when we talk about this. Sound is basically pressure waves, compressions and rarefactions in the air. And our brain interprets frequency as pitch and amplitude as loudness. So if there's a change in if there's a change in frequency, that changes the pitch that we interpret, and then also if there's a change in amplitude, that is also interpreted as how loud that sound is. And loudness is measured in decibels. And humans can hear from 20 to 20,000 hertz, with the best sensitivity being between 1,000 and 3,000 hertz. So that we have a huge range, but mostly like what we hear is in between 1 to 3,000 hertz. And anything above about 80 decibels for a long time can damage hearing. So that's why it's good to not listen to our music too loud in our headphones. So now that we understand the properties of sound, we can understand a little bit how sound is transmitted through the ear. So this is the first transduction. Sound waves hit this tympanic membrane right in here, which causes it to vibrate. So the sound waves come in and push it, which makes all of these, they're called ossicles, these bones in here, hit against each other and then push on this membrane right here, the oval window. And so as the vibrations pass through all these bones, the malleus, the incus, and the stapes, it pushes on this oval window and creates fluid waves in the cochlea, and that is the second transduction. So that's it for this slide. The next step is inside the cochlea. It looks like this seashell looking thing in here, and taking a slice out of that, this is what it looks like on the inside of all the squiggles. So the cochlea has 3 fluid-filled ducts in here, 1, 2, and 3. The vestibular, the cochlear duct, and then the tympanic duct. And so the vestibular and the tympanic ducts contain perilymph, while the cochlear duct contains endolymph, and endolymph is high in potassium, which will be important later. But inside is the organ of corti, which down here, if you zoom in, is this little thing. And this is really important to how we transduct sound into action potentials. And on here, there's hair cells that sit on the basilar membrane, and their stereocilia, the little hairs, touch the tectorial membrane, which is up here. This setup is vital to turning motion, the movement, into electrical signals. Yes, we'll go more into detail in the next slide. So these hair cells up here, this is where sound becomes electrical signals. So at rest, some channels are open a little, and then a neurotransmitter is released. So at rest, 10% of ion channels open. So there's low neurotransmitter release during that time. And then when these hair cells move to the right, towards the longest, I guess not necessarily to the right, but in this diagram it's to the right, these hair cells, follicles move towards the longest one, the stereocilia. The tip links pull channels open, so potassium and calcium rush in, and this causes depolarization, which causes more neurotransmitter firing. More neurotransmitter and more firing. And then when hair cells bend the other way, backwards, towards the shortest stereocilia, that is when channels close and it's hyperpolarization and less firing. And so this back and forth firing pattern matches the sound wave. And so that's how action potentials are made from the sound frequency. That it's just like our ear, the tympanic membrane, and then cochlear membrane, and then inside this is what happens with all those little hair follicles. So the next step in this is so like the part that happens in our brain. Pitch is coded by which part of the basilar membrane vibrates. So there's a high frequency near the oval window, which was hit by the stapes. Then, there's a, if it is perceived by the oval window, then it's a high frequency, and then if the further away it gets is a low frequency. So that is pitch, and then loudness is coded by the firing rate of the sensory neurons. So the louder the sound, the faster the firing. And signals leave the cochlea through the cochlear branch of cranial nerve 8. Let me see if it's on this diagram. Yes, these right here. So the signals leave the cochlea through this cranial nerve number 8, and they travel to the cochlear nuclei in the medulla, and then the midbrain to the thalamus, and then finally to the right and left auditory cortexi. And along the way, the brain compares the timing and input from both ears to figure out sound location. Isn't that crazy? That just blows my mind. Like, all of this is happening all the time, and we aren't even aware of it. It's just always happening, and I just think that's incredible. So in conclusion, hearing requires 5 transductions. First, the sound waves and the go to the tympanic membrane vibrations, and then those vibrations move the fluid waves in the cochlea, and then the fluid waves in the cochlea move the hair cell, bend the hair cells, which triggers ion channel changes. And then as ion channel changes, that leads to altered neurotransmitter release. And then the neurotransmitter release leads to action potentials in the auditory neurons. And I made a study aid for that to kind of summarize everything. Sorry, it's I should have made it a little bit bigger. Let's see, can I zoom in here? I don't know if I can zoom in, but maybe I can. Yes, so, okay, why is it doing that? Okay, so that is so this is the auditory transduction, which is mechanical to electrical energy. We see here for number 1, this is the sound waves hitting the tympanic membrane right here. And then this causes the eardrum to vibrate, which is in here. And then the tympanic membrane vibrations make the fluid waves in the cochlea. The ossicles amplify vibration. Stapes pushes on the oval window, which is right here. And then those vibrations become fluid waves inside the cochlea, which is in here. And then step 3, fluid waves bend the hair cells. And And then we jump over here to step 4. These are the hair cells that cause neurotransmitter release. And then that's just magnified, zoomed in here. And then step 5 is neurotransmitter and action potentials happening in the neurons. And then down here is just a little brief depiction. So the sound wave goes into the membrane, which is a mechanosensor, and then that leads to the ionic current, and then finally the action potential that is perceived in the brain. So that is the end of my presentation. I really enjoyed learning all of this as I was preparing, and I look forward to watching all of your videos as well. Thanks everyone. Have a good rest of your day.